Hello students, today we will study microbiology paper 1, general microbiology, in which we will study unit 3, unit 4 and unit 5. Unit 3 contains classification system in which we will study Whittaker classification and Carl Woolf's classification. In unit 4, we will study bacteria, its size, shape and arrangement of bacterial cells and its external to plasma membrane. In Unit 5, you will study bacterial growth and reproduction and traditional Indian concept of microbial growth and reproduction. So let's begin this presentation. Unit 3. So we will begin with classification system. What is classification system? Classification is the systematic arrangement of living organisms into groups based on the similarities and differences in their characteristics. In microbiology, classification helps in identification, naming, and understanding relationship, evolution, and practical applications of microorganisms. Ancient Indian concept of microbiology from Ayurveda and traditional texts. Long before the invention of microscope, ancient Indian scholars recognized the existence of invisible living agent responsible for disease and spoilage. In today's language, we see microorganism, and in ancient texts, we say that creamy or non-creamy. Key concept in Ayurveda. In ancient texts such as Charak Sahinta, Sushruta Sahinta, Atharveda, we can see a description of creamy as microorganism in these texts. Classification of creamy, it is generally based on its habitat, its origin, and effects on human health. Preventive and control measure, concept similar to sterilization and hygiene were practiced boiling water, exposure to sunlight, use of herbal disinfectant like tulsi, isolation of infected individuals. Let's start with Whittaker five kingdom classification proposed by R.H. Whittaker in 1969. This system classified organisms into five kingdoms based on cell structure, mode of nutrition, level of organization, reproduction, and evolutionary relationship. The five kingdom concept contain Monera, Protecta, Fungi, Monera protesta, fungi, algae, and plantae and animalia. So let's begin with Monera. Monera are the prokaryotic organism. They do not have uh, two nucleus. It includes bacteria and cyanobacteria as an example. They are unicellular, microscopic, and reproduced by binary fission. Example is the Shiaphila mycoplasma. Protista are eukaryotic, mostly unicellular organisms. They have two nucleus and membrane-bound organelles. Includes protista and unicellular organisms, ring worms. Example is amoeba, paramecium, rubina. These are the examples of protista. Then we come to the fungi. Fungi are also the eukaryotic and mostly multicellular organisms. They are made up of chitin and heterotropic and anthropic nutrition, and they reproduce as yeast mold and mushrooms. Plantae are the multicellular eukaryotic cell wall made up of cellular, tropic, and photosynthetic. Example is algae, morphs, fern, flowering plants. Animalia. Animalia is a multicellular eukaryotic concept heterotropic with ingestive mode of nutrition. Example, insect bird mammals. Significance clearly separates prokaryotic and eukaryotic and recognize fungi as in separate kingdom by the accepted of the classification system. Carl Rue's three-kingdom classification proposed in 1977 by Carl Rue, based on the 16S and 18S RNA sequence analysis. This system reflects evolutionary relationship model evolution in the classifications, molecular sequencing, RNA, genetic differences, evolution history, three domain, Archaea, eubacteria, eukarya. Archaea contains prokaryotic cell but generally distinct from bacteria. No bacteria like main cell wall often live in extreme environment. Examples are thermophile, halophiles, and methanogen. Methanogen produce methane. Eubacteria, two bacteria prokaryotic with peptidoglycan and cell wall. found in the all environment, and eukarya, all organism with eukarya cells. We can see the classification in detail. I can't zoom this image, but you can zoom this way. Three kingdoms and five kingdom classification. Classification of bacteria, which is actually the Burgi manuals of the systematic bacteriology. Burgi manual is a most authoritative reference for the bacterial classification and identification. It was published in 1923, which is based on the phenotypic and genotypic characteristics. Major classification criteria is methodology, staining characteristics like whether they are gram positive, gram negative, acid fast, and physiological and biochemical property, oxygen requirement, fermentation, and ability, and enzyme production, and major group of gram-positive bacteria, gram-negative bacteria, cyanobacteria, mycoplasma, and actinomycetes. Importance essential for microbial identification used in medicine and industry and research. Modern methods of bacterial technology. Molecular method 16 sRNA gene sequencing, chemotaxonomy, numerical taxonomy, phylogenetic analysis, and proteomic and metabolic approaches. Unit 4, we will study about the bacteria. This is the bacterial cell which contains its genetic material. This is the flagella. We can see the capsule and cytoplasm, ribocells, plasmids. We have villi also. Bacteria are unicellular prokaryotic microorganisms that lack a true nucleus and membrane-bound organelles. Despite their simple organization, bacterial cells possess a well-defined structure. They support survival, growth, and reproduction and adaptability in diverse environment. So let's begin with shape of bacteria. There are different type of shape. Cocci, bacillus, spiral, विप्रोज कॉमा शेप्ड होते हैं स्पाइरल शेप्ड होते हैं बेसिलर कॉर्ड शेप्ड होते हैं एलिमेंट ऑफ बैक्टीरियल सेल डिप्लोकोकस स्ट्रेप्टोकोकस स्टाइलोकोकस टेट्राट एंड सार्केटा these are the different types of arrangement of bacterial cells. Bacterial shape we can see in the image. There are the different type of bacterial spiral, rod shaped and cocci shaped. Structure external to plasma membrane. Cell wall present in almost all bacteria. Cell wall समय present होती है. Then we will study about the capsule. Capsule is a thick, very well organized, humanly attached to the cell, protect against the phagocytosis. Flagella. Flagella is the web-shaped structure responsible for the motility, made up of proteins. It's functioned for locomotion, chemostasis, and helps in the colonization. Fimbriae, short hair structures present in the large membrane and then stalk. प्रोटेस्टेड लॉन्ग ट्यूबुलर एक्सटेंशंस होते हैं सेल वॉल की या मेम्ब्रेन्स की स्ट्रक्चर इंटरनल टू सेल वॉल देन वी विल सी द प्लाज्मा मेम्ब्रेन जो कि सेलेक्टिवली permeable होती है then cytoplasmic innervation genome which is known as nucleoid and spores endosperms you know and cyst. In unit 5 we will study about the bacterial growth curve in which we can see that lag phase, log phase, stationary phase, and death phase. Bacterial growth means growth of bacteria. In bacteria, growth refers to an increase in the number of cells, not in the increase in cell size. Cell का size increase नहीं हो रहा है, but number of cell increase हो रहे हैं. Bacterial growth by grow by cell division leading to an exponential population increase under favorable condition. Logarithmic representation of bacterial population growth x-axis पे टाइम रहता है y-axis पे नंबर ऑफ वाइबल सेल्स होते हैं फेजेस हमने बात करी नेट फेज जहाँ पे क्या होता है नंबर ऑफ सेल्स जो होते हैं उसमें इंक्रीज नहीं आता सेल अडेप्ट टू न्यू एनवायरनमेंट एंड एक्टिव सिंथेसिस ऑफ एंजाइम्स प्रोटीन बना रहे हैं ड्यूरिंग दिस सेल वो अपने मीडियम पे डिपेंड करते हैं लॉग फेज जहाँ पे एक्सपोनेंशियल होता है रेपेरेंट काउंट एंड डिवीजन होता है पॉपुलेशन डबल हो जाती है ideal for the antibiotic sensitivity testing, industrial fermentation, physiological studies. The stationary phase rate of cell division equals to the cell death. Stationary phase में क्या होता है कि cell division का है rate वो cell death के equal हो जाता है and death phase पे तो सारे वाइबल सेल्स होते हैं वो डेथ की तरफ चले जाते हैं। हम उसे कैसे मेजर कर सकते हैं? टेक्निक्स फॉर मेजरमेंट ऑफ बैक्टीरियल ग्रोथ। So we have direct method, indirect method, diversity measurement, dry weight measurement, and direct microscopic count, variable count. Factors affecting bacterial growth, physical factor, temperature, pH of body pressure, radiation, and moisture. Reproduction in bacteria. Bacteria reproduce by sexual methods like binary fission, जिसमें क्या होता है, एक सेल रहता है फिर वो दो में डिवाइड होता है, फिर वो बहुत ही लीनियर डिस्टेंस से डिवाइड हो जाता है, फिर वो अपने through individual cells में divide हो जाता है. Budding or small outgrowth from one end of the parent cell but in larger than the reed. And then fragmentation जहाँ पे फिलामेंट में break होते हैं. Traditional Indian concept जहाँ पे हम क्रीमी की बात कर रहे थे. And the rapid multiplication is spread through contact air, water, and controlled nature of sunlight exposure. What I have. Thank you.